All right, good morning to my biology students. Mr. Clancy, I have some notes I want to go over with you today on DNA replication. I created some notes. Most of them I want you to follow along with me on the notes. I'd like you to watch the video a couple, three times to make sure you've got the notes. There's a lot of new pieces of information I want to go over with you. And I want you to give me your very best effort, okay, in going over with this. So these are the notes here we're going to be looking at for today. At the end, there is a short assessment I'd like you to take. I will post that in just as soon as I finish recording this video. I'll post that on, it will be on the assignment section. Make sure you do that today and send it back to me for a grade. Give it your best shot. There is an answer key at the very end. Don't peek too quickly. Try to do it yourself first. Really push yourself, and then see how you do with it. But basically today we're going to be talking about, you can see about DNA structure. We're going to be talking about and DNA replication. And then we'll go over the notes, and then there'll be an assessment. Most of these notes I've made out from before, just to make the video a little bit shorter. There is one segment that I'm going to draw for you, basically to show on the next page, the illustration, and how easy it is to draw DNA, specifically the nucleotides of DNA. You need to practice drawing these a couple of times to really get it down. But anyway, let's go ahead and go through it. We started this last week with a couple of videos, and then the work that's being submitted to me is very, very good. Make sure you stand up on your work, very important, not just in this subject, but in all your subjects. And I know it's difficult. It is difficult on all of us. Let's just do the best we can. I have your back. I'm going to be here to help you. I've had several students reaching out to me, so please continue to do that. Anyway, the structure of DNA is made up of building blocks, which are called nucleotides. All right, and each nucleotide has three components. One is called the phosphate group. Sometimes I will just dub that as P. The second is a sugar, and the third is a nitrogenous base. So I'm going to draw one, kind of looks for it. I'm actually going to start with the sugar. And the sugar basically would have an oxygen atom here. And then basically there's a this is a five-sided sugar, 5-carbon sugar, all right? And this sugar in DNA is actually called deoxyribose. You might remember that OSE always means sugar with that. Also, this is a carbon atom right here. This is the second carbon atom, a third carbon atom, a fourth carbon atom. Then there's a fifth carbon atom right here. And because of the positions of the carbon atoms on this sugar ring, this is called the 1 prime. Equals, the verbiage on that is 1 prime. The 1 prime carbon, this is called the 2 prime carbon. This one right here is called the 3 prime carbon. This is the 4 prime carbon. And this right here is the 5 prime carbon. And then typically, this is the sugar right here, the sugar. We'll just whoops. I wanted to keep that in black for you. This is the sugar. Over here on the left is going to be the phosphate group. And I'll just dub that as P. All right. And then I'm going to take this 1 prime out of here just for a minute. I'll put it up a little bit higher here. And then at the 1 prime spot is where your nitrogenous bases would actually be attached. So this nitrogenous base you learned last week, there are 4 different ones: adenine, thymine, cytosine, and guanine. All right. So and then they would actually be attached to, if this was this would actually be attached to another strand going on the other side. All right. And if you remember, adenine by Chargaff's rules always goes with thymine, always, 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 always. And cytosine always goes with guanine. All right. And I have an illustration I want to show to you. Now this shows, I just did a very short DNA strand here. This is the DNA with it. And you can, a couple of things I want to show to you. This would be 1 nucleotide right here. This is a second nucleotide. a third nucleotide, and fourth nucleotide. You might notice that the pairs on the right here are upside down. And that's, I'm not going crazy, that's by design. These strands are what they call anti-parallel. In other words, they run in actually opposite directions. Each new nucleotide is actually added onto the 3' at the 3' spot. And you'll see, as we'll talk about in just a minute, replication always occurs in a 5' to 3' direction. So replication would actually go this way. All right, I'll come back to that in a few minutes. The reason I put this 3' to 5' is to show you the direction. Let me explain what I mean by that. So again, this would be one strand of DNA. This would be the other. When DNA replicates, these bonds between what we call the complementary bases are going to get cut, as I'm trying to show here with it. And then you create basically two separate strands. You create two single strands from the one double strand, and each single strand acts as a template for the new strand of DNA. So this is amazing, and it's all because of these complementary base pairs where the adenine matches up with the thymine and the cytosine with the guanine, that you always get an exact replicate, with rare exception. There are some exceptions to that, but more on that later. But basically, every time we make new cells, we need our own DNA in that cell every time, and this allows the way this law of complementary base pairs allows us to get an exact strand. So some other notes. So I want to go through this, then I want you to try the assessment as we go through it. The two strands of DNA, just follow. The two strands of DNA move in opposite directions, as we mentioned earlier. All right, so this one's moving, you can see, in this direction. All right, and you can see how they're actually opposite directions with it. Now, DNA replication, as I mentioned, moves in a 5' to 3' direction. And these numbers, as I indicated earlier, correspond to the carbon atoms on the position of the sugar molecule. All right. Now, you might ask, how do I know which way is 5' to 3'? And the answer is you look at the oxygen atom. Where's the oxygen atom position? So if I go back to my illustration here with it, you can see the oxygen atom is right here at 12 o'clock. Here it's on the other strand, it's at about 6 o'clock. So you look where the oxygen atom is present, and that you're going from a 3' to 5' direction. In this case here, on the other side, this strand goes down from 3' to 5'. So the 5' is basically going in the direction of the oxygen. Real easy way to remember which is which. All right. All right. The third one here is 3' to 5' direction. Number 4, the 3' spot, if you will, is where the next nucleotide is added. So this is your 3' spot right here. And that's where the next nucleotide is added. Remember, the replication will occur in a 5' to 3' direction. So the DNA replication is going to go actually this way. All right. That's number 4. Number 5, an enzyme called DNA polymerase. Remember, enzymes end in ASE. DNA polymerase is the enzyme that's responsible for adding the two nucleotides onto each strand. That's going to move in a 3' to 5' direction. So this is moving opposite in the way that the nucleotides are actually added on. One way to think about this is if you imagine going into, let's say, a movie theater, and you bought your ticket, and then you're going through the turnstile, right? There's a man there or a woman there to collect your ticket. They are facing opposite you. All right, so you're going toward the movie theater, and they're actually facing opposite you and taking your tickets. So basically, DNA, and I'll show this to you on the illustration in just a few minutes that I have up here. All right, in this case here, this is the 5' direction right here, and this is the 3'. Okay, so in effect, replication is going to go this way. All right, and the DNA polymerase is moving in the opposite direction. For this strand down here, All right, the replication is going this way, and notice DNA polymerase is actually moving the other way for that. And I'll explain some of these other terms in just a few moments. So DNA polymerase kind of gives out in effect, is adding the nucleotides onto each strand. The DNA replication, as we know, takes place in the S portion of interphase. And then some DNA roles I want to share with you as we go through this. There's one called gyrase, and you've got to go through this illustration now to really get this, but gyrase assists in unwinding the DNA. The DNA is like a double helix, like a spiral staircase, and when we unwind it, we basically go from a spiral staircase to like a ladder. The helicase, the second one, the helicase actually is basically I call it the scissors. It actually cuts the bonds. These are hydrogen bonds between the nitrogenous bases. So these bonds are firm enough to hold the bases together, but they're not so firm that they can't be broken when they need to be for replication and later on for developing proteins, as we'll see. There's an enzyme called RNA primase that basically, you can see it synthesizes a short RNA primer. If you've ever had to prime a pump or kind of get something going, you'll understand what priming means. If not, just remember it adds the first nucleotide. Okay, so that's the role of RNA primase. DNA polymerase 3, as I mentioned earlier, adds the new nucleotides to the old strand in a 5' to 3' direction. We'll go over that picture in just a minute. And then there's something called DNA polymerase 1, which will replace the RNA primer with DNA. So that's sort of like a cleaning up enzyme, if you will, kind of takes care of business with the RNA primase and that RNA primer that made it before. And then there's one last enzyme that I want to share with you, which is called DNA ligase. And this joins what we call something called Okazaki fragments. Kind of a cool name together. So it makes a continuous strand. So I'm sure what's going through your mind is, Mr. C, this is really complicated. What are Okazaki fragments? And the answer is in this illustration. If I could show this to you. All right, so right here, you can see the gyrase is doing the unwinding, right? It's unwinding the strand of DNA. The helicase is cutting, cut. This is the scissors. Scissors that basically cuts those hydrogen bonds. And now I have two strands. I have one strand, which is called the leading strand. And there's another one called the lagging strand, which lags behind the leading strand. So you might be thinking, why does the lagging strand lag behind the leading strand? And the answer is the direction in which the DNA is replicated. So what this is doing, this strand is going to unwind, and the replication is basically going in this direction. So on the leading strand, the replication goes this way. All right, so it's basically, there's the replication occurring. On the lagging strand, the replication is going this way. Replication. Oops. See if I can I-O-N. All right, so replication going this way, but if you'll notice, this replication up here is going away from this area here, which is called the replication fork. So as replication on the lagging strand, or the leading strand is going away from the fork, it's actually going to the fork here. And there are times when the replication, it kind of gets temporarily set back a little bit because there's not enough of the single strand unwound in order to replicate, in order to add new nucleotides onto. So there are some pauses, if you will, a break in the action, if you will, where the strand, there has to be a little bit of waiting time for more of the single strand to be cut and unwound. So during that particular waiting time, there are no nucleotides added in, and there are gaps. All right, there are actually gaps in these in the new strand of DNA that's made. So they're not temporarily, they're not continuous. They're actually discrete. These gaps, these fragments of DNA, all right, are actually called Okazaki fragments. All right, so this is fragmented DNA on the lagging strand. So it's lagging behind the leading strand. In order to bring these together, there is an enzyme which is called ligase, DNA ligase, kind of acts like a glue and brings the Okazaki fragments together. So you end up having a one continuous strand of DNA. So that explains about the leading strand, which sort of replicates free because it's going away from the fork, and the lagging strand is going to be fragmented replication because it's going toward the fork, and there are times when there's not enough a template DNA strand, if you will, to work on. So that's today's lesson. Make sure you go through it a couple times. Keep it in your records for later study, which you may use on some assessments. And looking forward to seeing you. We'll just keep praying that this period of confinement will end in the not too distant future. And just remember, we have a God who is greater than the coronavirus. So, all right, all the best to you guys. I will chat with you later. Have a great day.